welcome you to the course on neural science for engineers. This is Srinivas Bhaskara, TA of this course, and today we are going to see how brain stimulation concept works and what are the different things that are involved in brain stimulation. First we will see what is a brain stimulation, then why people are doing brain stimulation, what are the different research perspectives in terms of brain stimulation, and please be clear that this we are not going to deal in the biological aspects and clinical aspects of brain stimulation. We will restrict ourselves to engineering perspective of brain stimulation, like how to fabricate a device and what are the currents that are involved in this, or what are the circuits that are required to stimulate that particular device, and what are the materials, what kind of materials we need to choose, something like that. Okay. So, we are not discussing anything about biological aspects like what kind of transmitters are generated or destroyed or in the process or whatever it is. Okay. So, we restrict ourselves to engineering perspective. Okay. So, let us get started. All right. So, if you see the slide, what is generally a brain stimulation, like controlling abnormality in a particular brain region by applying electrical signals, right. So, let us understand what is this abnormality. What I mean by that is, let us say I wanted to move a hand. Okay. So, I think that I want to move a hand, like thought will be created and then it will create some kind of impact in the brain. Okay. And we, by using some imaging techniques, people can see which part of the brain has an impact by thinking something or by sensing something kind of thing, right. So, if I want to move my hand, I can move my hand like this. Okay. But if there is abnormality in the brain, something has caused the brain to malfunction or some abnormality is there, like it is not working normal. That kind of situations, what may happen is, I wanted to move my hand like this, but because there is abnormality and the transmission of the signals are not happening properly. So, what would happen, end up happening is I will be like this. Okay. It will be like this, something like that. Okay. It is not proper, you see my fingers are shaking. Okay. It is like that. Okay. So, this is kind of abnormality, whatever I wanted to do particular action, okay, I am not able to do it. This is kind of abnormality. So, what are the reasons for abnormality and all? There are so many things. Okay. We may see some of them, right, and how we can treat it. Okay. That is the next thing. Okay. So, if you look at my slide here again, there are, this is one thing is abnormalities in particular brain region, okay, and clinically accepted. So, to treat it, there are clinically accepted surgical treatments are already available. Okay. And if you see, there are like many neurological disorders like Parkinson's disease, essential tremor, dystonia, and epilepsy, obsessive compulsive disorder. There are many such neurological disorders exist, and through brain stimulation we are going to to get back to the normality of the brain, but let us see whether we get back or whether completely we can get it back or partially or something like that. So, the idea is you apply that particular electrical signals, okay, to that particular region. Let us say for example, I may be discussing more about subthalamic nucleus of I mean not in terms of anatomy, I may be using this word subthalamic nucleus, or I may be referring to this sometimes in the course of our study. So, there is a region called subthalamic nucleus, make a note of this. This region is more or less nowadays presently used for treating people with Parkinson disease. Okay. So, we may be discussing more about Parkinson disease as well. Okay. We will take up one example and then see how people are doing brain stimulation. Okay. And what we can do is see, if you see, look at the brain. Okay. This is the top surface, let us say this is the surface of the brain, and maybe whatever the deep inside these structures, okay, these structures like subthalamic nucleus, thalamus, all other things, okay, they are something called as a deep brain structures, let us say for example. Let us understand deep brain structures. Okay. So, there are devices okay, which can stimulate the only the top surface. Okay. I can also call it as a cortical surface, right. See, once again I am telling, we are not discussing the anatomy and all, this is for just for understanding, so that we can understand this thing in a better way. Okay. So, we can stimulate either cortical surface or we can also stimulate deep brain structures, depending on what kind of applications. Okay. So, for a Parkinson, okay, you can have something called as a motor cortex, okay, and you have something called as subthalamic nucleus. It also stands, so STN stands for subthalamic nucleus. Okay. So, these regions are so much of important. Okay. You can stimulate, but motor cortex generally will be on the surface. Okay. Right. So, you can refer to the anatomy of a brain and then you can see what is, where is the motor cortex located and all those things. Okay. Our discussion is not about the biology. So, I am saying these two regions are important. Okay. So, people can stimulate motor cortex as well as subthalamic nucleus to see to see whether we can regain back the normality. Okay. So, now there is other important region is called PPTG region, okay, peduncopontine tegmental nucleus. Okay. So, I don't expect you to remember all these names and all, but I am just telling these are the names that are referred to. Okay. So, the targets, these are mainly important targets for Parkinson disease and all. So, and for a Parkinson disease, the origin of the treatment started from 1980s, means you can say roughly some 4 decades back. Okay. So, if you want to go through, you can just I have given the references here. So, you can just refer to them and then you can see what kind of work that has been done so far. Okay. So, now we are very clear the ideal the situation is here, okay, like this. So, we understood what is brain stimulation. Okay. You are stimulating the particular region in the brain by using electrical signals. So, these electrical signals, how we are going to supply and what kind of circuits that are involved and all those things and how to supply, that we will discuss. Okay. So, the next slide, if you see, why brain stimulation? Okay. So, generally people may think, why do we need to stimulate a brain, correct. So, generally for a neurological disorder, initially it is treated by using a medication or a diet, right, that is at the primary stage. And this is important, this DBS stands for, okay, deep brain stimulation. Okay. So, DBS stands for deep brain stimulation. If somebody does not respond to the medication, then the other therapy that is available is a deep brain stimulation, means you open the brain and put the devices that are required and you choose that particular location. I already told you subthalamic nucleus or whatever the interest is. Okay. It is not the discussion about which region is. My idea is to tell you that you just opening the skull and then opening the brain, I mean exposing the brain. Okay. You cannot open the brain, you just expose the brain and you place the devices wherever the region that is required and supply the electrical signals and then stimulate it, and the people has got the benefit out of it, right. So, that is why it is most research topic right now, right. So, and you look at the numbers and you look at the slide, there are almost 10 million people, they are estimated to be affected, and we do not know how many else, how many other people are also there, and these are the general symptoms of Parkinson disease. Why it is important is, I told you about motor cortex, okay, already. Okay. The name itself says that there is something called motor. Motor means like movements. Okay. In a layman terms, I am saying this is for engineers, right. So, whatever is related to the movements disorder, okay, may arise, I am saying may, okay, arise from the motor cortex, but the main source could be somewhere else like STN, and it may pass through motor cortex or something like that, right. So, the idea is whenever there are kinds of whenever there are movements that happens, whenever we are moving the hands and all those things, right. So, the cortex region through imaging techniques, what people have found out is, okay, the region where most activity takes place in the brain. Okay, so let us say that we call it as a motor cortex, means we may say that if you move the hand, okay, or if you turn right or left or something like that, okay, some kind of motor action, like some kind of movement takes place in your body. Okay. So, that means that there is some activity that is happening in the brain. So, that region, let us say, call it as a, we just refer to as a motor cortex, let us say, for example. Okay. So, if there is any disorder, okay, then we have to deal with the motor cortex region. Okay. So, these are the different symptoms, and like tremors, sloshing, loss of balance, trouble in swallowing, limbs are very stiff and they are not proper to fold or something like that, and trouble in walking, mask like expressions, very difficulty in moving the muscles of the face, and trouble in sleeping also. And these are the some of the symptoms, but there are many other symptoms. There should be a trained neurosurgeon who has to qualify the patient to be a Parkinson disease or not, and what kind of treatment is suggested to them. Okay. So, if you see the next slide, how the deep brain stimulation is done. Okay. So, there is something here, if you look at this part, okay, this is a, let us say, pulse generator. Okay. So, what is the idea? The idea is you have to send electrical signals, right, to the brain. This is the idea, correct. So, now the neurosurgery has been performed on the brain, and they would have opened the skull and they would have exposed the brain, and using some stereotaxic operators or any other surgical operator that is that is available, they would have understood that they would have already placed this device. Okay. Because we are not talking about surgery and all, we assume that everything surgery is taken place, and then you have inserted this device, or you can say implanted. Okay. Right. So, you have implanted the device. Now, you see this, okay, the device structure looks like this. Okay. There is some kind of cylindrical shape. Okay. Now, I will also tell you why cylindrical and all those things when we discuss further. Okay. And there are some kind of dots here, you can see, okay, or rectangular pads, right. So, that you can see. So, those rectangular pads are like this. Okay. So, these are nothing but a contact pads. Okay. All these four, all these are contact pads, and internally there is a wires. Okay. Right. Internally there is a wire. Of course, all the four are not short circuited. Okay. Understand that. Okay. Otherwise, it is not going to work. So, there are four contact pads that we can see, and internally there are wires, and here there is a connector. Somehow all the four contacts are taken like this, okay, through a wire, and understand that all the four contact pads are available. These are contacts, and there is something called this, you can say, contacts. Okay. And there is something also called as contact pads. Okay. Because how you need to supply the current through this, I mean how you need to supply the current to the electrodes? By having some contact pads, okay, somewhere at the end. Now, these contact pads will go to the circuit. Okay. So, you have the contacts here, okay, distinct four different contacts like this, and you have the wires that are connecting them to the electrical circuit. Okay. Let us say this is a pulse generator, I can also say circuit. Okay. Now, that is how the connection is established. Understand that I am just trying to explain how the things work here. Okay. So, now this pulse generator has some electronics that are inside, okay, and you can generate the waveforms, okay, whatever the electrical signals that you want, we can generate like that. And okay, this kind of signals we can generate. This is nothing but a biphasic pulse. right. If you look at this, you have positive side of the, it can be current or voltage whatever it is, we will discuss about that later also. You have negative side also, let us say this is 0, okay, this level is 0, right, and you have equal positive amplitude and negative amplitudes of current or voltage whatever it is, okay, and this is this is they call it as biphasic pulse, and this is the preferred one. Okay. So, you have monophasic also. So, monophasic looks like this, okay, just like this, okay, this is monophasic, correct. So, the idea behind this biphasic is whenever you stimulate something, whenever you send this positive signal, right, or positive voltage, for example, understand like that in a layman terms I can just explain. So, when you send a positive voltage, what happens is there will be a, you are transferring the charge, correct, you are transferring the positive charge and you are retrieving a, I mean you are transferring the positive charge, that means that you are sending some extra charge, that is means the brain is already there and you are transferring some extra charge to that, right. So, that extra charge should not damage the electrolyte that is present in the brain, right. So, what you do in the next cycle, okay, you retrieve this, I mean in the same cycle only, this is the cycle, let us say I call it as one cycle. Okay. So, during that cycle, okay, you send the positive charge and you retrieve the same positive charge, that means in terms of like this negative, correct. So, that you are having something called as charge balance, charge balance. Okay. So, the basic idea is you are not sending extra charges to the brain electrolyte, okay. You are stimulating, you are sending it and again you are retrieving it back, okay. And in this way of doing it, the people have observed that there is a normality that is maintained from abnormal nature, means you are there are some deactive neuronal region, something like that, it would have got normal or activated, something like that, okay. So, that is the effect that is felt, right. So, this is how generally a deep brain stimulation thing works, right. Let us see the, now what kind of electrode material I can choose, what kind of substrate material I can choose. Okay. Let me tell you one thing, okay. I have drawn that cylindrical structure like this here, right, and there are contact pads, right, and there are wires that will go, okay, without any short circuiting it with each other. Okay. There is a whole lot of engineering that is involved in fabricating this device. Now, only this part whatever I am highlighting with the red, right, only this part is contact, okay, mean this part only can stimulate, right. Okay. Let me write it here, only this part can stimulate, only this part can stimulate. The white color region whatever is present in between these two, contact 1 and 2, okay. So, that is a insulator, right. This whatever the white space that is present between the contact 1 and contact 2 is just an insulator, right. So, only that part is a electrode, okay, that will act as an electrode, right. Now, whatever the body, okay, that is containing all these electrodes, okay, this entire structure, let us say we can call it as a substrate, okay, the material that is supporting. So, this substrate will not conduct anything, okay. So, it is like an insulator, right, only the electrodes will contact, are you getting it, right. So, this is how the structures are, but what kind of materials that I can use, or what is the criteria, is there any criteria I can, or I can choose randomly whatever I want. So, the answer is yes, there is a criteria for choosing the electrode and the substrate materials. So, the first criteria is, you need to understand this, this is just, see this is not just a electrical system, okay, and then you just give a electrical circuit connection and then the electrodes will start generating the current or voltage whatever is required. Okay. So, it is implantable device, you have to implant it finally, right. Where you are implanting it? You are implanting it in the human brain, okay, it is in the body. Okay. So, you know this something called as immune response and all those things, whatever that comes from outside the human body, the brain sees it as a foreign body. Are you getting? That is very clear no, for all of us, we understand no. So, look at the slide, the first criteria is this: whenever you implant anything, there is something called as some kind of foreign body response is there. Okay. That foreign body response will try to, I mean, because of that there is something called as scar tissue formation around the implant, okay, and there will be deterioration also that takes place if the foreign body response is too much, right. Suppose if you want to use your electrodes inside the body, this has to be ideally, okay, it should be there is no zero, there is no response at all, or if not possible minimal, as minimum as possible, right. So, there is a whole lot of research going on developing the materials, okay, based on this, and the second requirement is the biocompatibility. Okay. So, whatever the material that you are putting it inside the brain or inside the body should be compatible, okay, and what it should not do, it should not cause toxic, allergic or other harmful effects, right. So, for example, some of the biocompatible materials I can give platinum, okay, gold, titanium, right, which we are known, I mean I am talking about material metals only, okay, like electrode things and all I am talking about. And some of the other things are like you know there is something like silver, and there is something called as and there is a debatable things are also there, okay, in that. So, there are other materials which are not biocompatible. So, you have to make sure that they comes under the biocompatible category, and there are whole lot of studies to decide whether the metals are I mean whether the material is biocompatible or not, okay. And the another requirement is it should not generate any kind of chemical products or byproducts, means you have inserted in the brain, you have inserted the device in the brain, it should not react with the brain whatever the materials that are present inside the brain, and then when what happens when it reacts with this, then of course, there is a byproduct, and because of that byproducts and all it may cause another effect on the brain, or because of that there is a degradation that happens in the brain, I mean degradation of the electrode material may takes place or substrate material can takes place. So, these are the minimum, these are the basic requirements of choosing a electrode and a substrate material both. Whatever if it is inside, first thing is it should create a very minimal foreign body response, then second thing is it should be biocompatible both electrode as well as substrate material, okay, and it should not generate these chemical products or byproducts as a part of the reaction, right. So, next these are the basic requirements. The other things are there is something called as soft versus hard, okay, implants. What does it mean? Okay. So, if you take any material, it has some kind of stiffness, I can say it may have something we can characterize using something called as Young's modulus or something like that, right. So, the brain, if you see the brain tissue, the Young's modulus of the brain tissues, okay, for example, I am just giving some number, do not quote me on that, they in the range of kilopascals, okay, in the range of 3 kilopascal or something like that, okay, for if you take for example any brain tissue of a rat or something like that, okay, in terms of kilopascals, right. So, but if you take silicon for example, because most of the time we use silicon as a substrate material okay, where on top of that we are going to fabricate these electrodes according to our geometry requirements and all those things, right. Now, if you talk about silicon or silicon dioxide, something like that, okay, their Young's modulus is in the range of gigapascals. Okay. I can give you some numbers like it is around 140 gigapascal or something like that in that range, silicon, right. So, what may happen is when you are going to, it is very simple, you have a I can show you like this, okay. You have a smooth surface and it is very smooth and you have very hard material, and if you poke it, okay, what will happen? It is going to rupture all over, okay. It is, and you are ending up damaging it. Not only that, why it is so important is mechanical property, this is I can say mechanical property. Why it is so important is there is something called as micro motion of a brain, okay. Your brain will be, it will not be stable like this, like a stable thing, right. It will be keep on vibrating. If you can see my movement of the hand, it will be like that, somewhere like that, and you remember now you can understand, okay. Now you are putting, this is a soft substance, my hand, let us say this is a brain, and you are putting an implant, okay, which is hard implant like a silicon or kind of thing, okay, and this is moving, what will happen? Okay. This is going to, this implant is going to rupture the brain, and this causes something called as local irritations, right. And because of that there is a local irritation, and your brain will start doing whatever it has to do, like it has to recover. It is like you have something called immune response, right. So, wherever there is a damage in the brain, there is something called, not only in the brain, wherever in the body there is something called immune response that is triggered, and this immune response is going to protect the brain, okay, and there is a local irritation is happening, these things are happening parallelly. So, what will happen? There will be a you may not get a perfect contact of this particular electrode material with the brain, okay. Means there is lot of things that are involved in, because of the immune response there is lot of other things that are formed between the electrode and the tissue, correct. So, that is why whenever you choose an implant, you have to choose in such a way that it should be as soft as possible, and it should be compatible within the ranges of the brain, okay. So, let me give some examples. Now, hard I already explained, you have something called a silicon wafer. If you take a silicon wafer that considers as a hard implant, I mean I am saying their Young's modulus is in the range of gigapascal, and there is something called SOI, okay, silicon on insulator wafers, okay. Then let us talk about soft, okay. So, you have something called as polyamide, we can call them as PI short form, okay, polyamide, and there is something called parylene C, and there is something called as, okay, these two examples are sufficient to understand, okay. These are very much compatible with the brain tissue, when compared with the silicon and silicon wafer, SOI wafer, are getting it. So, there is lot of research that is also taking place in terms of inventing or discovering the softer implants, right, and this is comes under mechanical aspect, right. So, the another one which is most important is, let me erase everything, right. These are electrical properties, right. So, finally, these are related to the electrode, okay, to the electrode. So, we have seen what are the different criteria of choosing electrode material, and one thing is the most important thing is the electrical properties. What are the things to be considered for electrode also? I mean what are different properties that we need to look into it, that we will see in the next lecture. Thank you.